In a limestone cave in the Altai Mountains of southern Siberia, researchers pull a bone fragment smaller than a fingernail from sediment picked through for decades. It is a sliver from the tip of a child's pinky finger, one of thousands of unremarkable fragments recovered from that cave since the 1970s. Nothing about it looks significant. No tool marks, no ornament, nothing to separate it from the pile. For 2 years, it sits cataloged in storage before anyone decides it might be worth testing. Then, in a laboratory in Leipzig, Germany, a team specializing in degraded ancient DNA extracts what survives inside it and sequences the result. What comes back does not match a Neanderthal genome. It does not match a modern human genome either. It matches no population anyone studying human origins had previously identified, named, or expected to find. It was less a discovery than a problem. The data did not fit anywhere on the tree that a century of paleontology had built. For most of the 20th century, the story of human origins in Eurasia had two characters: Homo sapiens, who evolved in Africa and began moving outward roughly 60,000 years ago, and Neanderthals, a cold-adapted cousin species already established across Europe and Western Asia, first identified from a skeleton found in Germany's Neander Valley in 1856. The two overlapped for millennia before Neanderthals vanished from the fossil record around 40,000 years ago, leaving modern humans to continue alone. It was a clean, two-branch picture, reinforced by decades of skeletal analysis and by the first successful sequencing of Neanderthal mitochondrial DNA in 1997, led by a young geneticist named Svante Pääbo. Textbooks drew the family tree with exactly two lines converging: us and them. Asia, in this picture, was largely empty of any parallel story. Whatever archaic populations had once lived across that vast stretch of continent were assumed to have vanished so early and completely that they left nothing behind for science to find. The finger bone changed that. When mitochondrial results came back in 2010, followed by a full nuclear genome later that year, Pääbo's team, working with population geneticist David Reich, confirmed the fragment belonged to a young female from a population genetically distinct from both Neanderthals and modern humans, a sister lineage that had split off and continued on its own path for hundreds of thousands of years. Researchers named the group Denisovans after the cave. It helps to understand what this kind of sequencing does. It does not guess at appearance from bone shape. It reads inherited molecular sequence directly, letter by letter, against every known reference population. This was not a matter of interpretation. The genetic distance was real, measurable, and could not be explained away as an unusual Neanderthal variant. A handful of teeth and bone fragments found in the same cave in later years belonged to this same population. An entire branch of the human family, unknown to science, had been identified almost entirely through molecules rather than bone. The obvious next question was where this population's legacy had gone, and the search produced a genuine puzzle. When researchers scanned modern genomes for Denisovan ancestry, they found it distributed with an unevenness nobody had predicted. Populations in New Guinea, Aboriginal Australia, and the Philippines carry roughly 4 to 6% Denisovan DNA, among the highest archaic ancestry percentages found anywhere on Earth. Mainland East Asians and Native Americans carry a real but far smaller share, often estimated near 0.2%. Europeans and most African populations carry essentially none. One early explanation proposed a single ancient mixing event followed by dilution as Denisovan ancestry spread thin across expanding, intermixing populations. It was a tidy model. It also ran into a direct problem. Tianyuan man, a 40,000-year-old skeleton from China, already carried Denisovan ancestry at levels comparable to living East Asians today, meaning the low mainland signal was not later dilution. It had been that low from very early on. A second explanation fit the data better, not one admixture event, but several, from more than one distinct Denisovan population. A 2018 analysis of Han Chinese, Japanese, and Dai genomes found evidence of two separate Denisovan-related contributions, one resembling the ancestry carried by Papuan populations, and one resembling the single high-quality genome recovered from Denisova cave itself. A follow-up study of Papuan genomes in 2019 pushed the number further, identifying signals consistent with several deeply divergent Denisovan lineages, separated from one another for a very long time before any of them ever met a modern human. This fits the unevenness better than a single dilution event does. One proposal suggests that ancestors of Papuans crossed the Wallace Line, the deep water boundary separating mainland Asia from Oceania, and met Denisovan populations there directly, while mainland groups only ever brushed against the edges. But no model fully resolves why the gap is quite this wide. The deepest answer to why any of this still matters arrived in 2014 from a study led by geneticist Emilia Huerta Sanchez. Tibetans live permanently in the thin air of the high plateau, where oxygen is scarce enough to cause altitude sickness in unadapted visitors, and they do it without the thickened blood that usually accompanies high-altitude life elsewhere, a trait long tied to a gene called EPAS1. When Huerta Sanchez's team resequenced that region in Tibetan and Han Chinese individuals, they found a haplotype at EPAS1 essentially absent everywhere else in modern humans, rare even among Han Chinese, and matched almost exactly by the Denisovan genome itself. Modern humans had inherited a working solution to high-altitude survival directly from a population that no longer physically exists, adapted for a challenge modern humans had not yet faced when they first left Africa. It was not the only such gift. Later research has traced Denisovan-derived variants near the OAS gene cluster, shaping antiviral immune response in Papuan populations today. There is a kind of inheritance that has nothing to do with money or memory. Imagine discovering that a skill keeping you alive right now, something as basic as how efficiently your blood carries oxygen, was never really yours to begin with. It was handed down silently by a relative you will never meet, whose face no one alive has ever seen, across a gap of many thousands of generations. You did not learn this skill. You did not earn it. It was folded into your cells before you were born, waiting for the one circumstance, a mountain plateau, a thin-aired village, where it would finally matter. That is what a Tibetan child inherits from the Denisovans without ever hearing the name. Most of us carry some version of this, fragments of instruction written by populations that no longer exist, tested across hundreds of thousands of years of trial and death, quietly folded into our own genomes. We do not think of ourselves as archives. In the most literal molecular sense, that is exactly what we are. None of this diminishes what it means to be human. If anything, it enlarges it. The two-branch family tree felt tidy because it was incomplete, and the discomfort some researchers felt when the Denisovan data arrived was really discomfort with how much more layered the true picture was always going to be. Denisovans themselves were not one simple population either. Their genome carries faint traces of mixing with an even older, still unnamed archaic group, sometimes called a super-archaic lineage, possibly descended from populations that had left Africa nearly 2 million years earlier and separated from everyone else long before Denisovans existed. Absorption into another lineage's genome was never uniquely our story. It happened to them too, long before it happened to us. Every population, living or extinct, seems to be built from older populations, folded into newer ones, again and again. Identity, at the genetic level, was never a single fixed point of origin. It was always layered, accumulated, ongoing, still being written, still being read. For 15 years, Denisovans remained a population without a face, identified by genome but almost no skeleton anyone could look at. That changed in 2025. A skull found in 1933 near Harbin, China, hidden down a well for decades to keep it from wartime confiscation, had been described in 2021 as a possible new species nicknamed Dragon Man. Its bone DNA had degraded past recovery, so geneticist Xiaomei Fu's team tried something almost nobody expected to work: scraping ancient plaque from the skull's teeth. Inside that overlooked residue was enough mitochondrial DNA to confirm what the bone alone could not. Dragon Man was a Denisovan, the most complete one ever found. Around the same time, a jawbone dredged from the seabed off Taiwan was confirmed by protein analysis as a second Denisovan, this one from a warm coastline, rather than frozen mountains. A population once defined by absence now has a face, a range, and still more questions than answers. Perhaps that is what real history looks like once you finally read it closely.